Okay, this is lecture 20, which I think is going to be relatively short for the prep. We'll finish mouse development. I'll talk about gastrulation. I'm not going to talk about turning, and so we'll leave the mouse embryo at the end of the gastrulation process. And then I'll just talk a little bit about human development. A lot of it is very similar to the mouse, but I'll try to remember to point out the places where there is difference. And to be very honest, is the type of development I know the least about. And I think that's probably true of many people because we don't study it in the kind of depth that we do the other organisms. Okay, so we basically finished looking at the implantation process. And implantation takes place around 4.5 days. And then you have this, let me just get the pen going. You have this egg cylinder. It's referred to as an egg cylinder. And as I mentioned, the early embryonic cup shape is unusual. It's confined to rodents. And so the beginning of gastrulation is around 6.5 days. And this is the first kind of implication in a posterior to anterior axis. And so on the posterior side, there is a thickening of cells. So when you look at this, instead of being columnar and sort of a single layer of cells, there's multiple layers here. And this is the initiation of the primitive streak. In the case of, I think I mentioned this already, in the case of the mouse embryo, it doesn't form a groove or a furrow the way it does on the chick embryo. Instead, it is this thickening of cells, and the cells will extend it starts at the posterior and works towards the anterior, just like the primitive streak in the chick embryo. So I'll just get this random thing out of here. So this is, they're referred to as a condensation of cells because it's a kind of a thickening. And it takes a half a day to a day to elongate towards the anterior. And when it reaches the bottom of the cup, so it doesn't come all the way around, but it ends up in the anterior. And just notice that this is dorsal in this embryo, and this is ventral. But if this was ventral all the way around, and this was dorsal all the way around, it is a really strange organization compared to what we're used to with other organisms. Okay, so that's the formation of, there will be a node that develops here. And unlike the chick, it doesn't kind of move back. In fact, it stays in place and the cells move through it. Oh. So this shows this is the kind of the thickened cells down the posterior, and it shows them sort of in progress moving through. The epiblast is this part, and so there's a couple of things that start happening. One is the extension and movement of the epiblast cells around and internal, and they contribute to the conglomeration of cells that is forming this primitive streak. But also, once this is formed, some of the cells here, they start migrating inwardly, and they find themselves, they place themselves between these epiblast cells and the endoderm that's on the outside. So they're sliding themselves in between these two layers, and they're going to be forming a mesoderm layer. Eventually, the primitive streak reaches kind of the base of the cup, and then uh, a set of cells there forms what is called the node. Um, but just like the Henson's node of the chick, this isn't a structure. This is more a gene expression, right? So what happens is you have migration of cells from posterior through to anterior, and as they move through the node region, their gene expression alters. So again, the node is less a structure and more a communication and organizing center. As the cells move forward, the mesoderm are going to form the notochord, and the ectoderm cells are going to form what is called the head process. So um, that's a structure that kind of extends anterior and is going to form the neural folds and so on. And when that folds around to form a tube, it gets elaborated to form the brain. And we probably will talk a little bit about brain formation, but later in the semester after the midterm. Okay, so this is a different figure, but if you take this structure um, and then you kind of pick away the outer layers and just look at like all the stuff that's on the outside and just slice through, then this shows you schematically approximately what it looks like. It's kind of blurry. I wonder why. And so here on the outside is there's ectoderm. There is this is the neuroectoderm. This is a head process. Did I say ectoderm? I meant endoderm, the yellow on the outside. And there is the node here, but there's also other mesoderm that's going to form somites and so on eventually. And the amniotic cavity is forming here. And as I mentioned in the previous lecture, the amniotic cavity is enlarged with programmed cell death. So this is a form of cell death that is directed. There are lots of embryonic processes that require programmed cell death. So the cells between your fingers, when your little hands form as an embryo, they form as kind of a pad of tissue, and then the cells between the fingers die away to give you unwebbed fingers, just as an example. And in neural development, many more neurons are produced than are going to be needed. And after synaptic connections and so on have taken place, the ones that are excess to purpose, they're no longer needed, those are also they undergo programmed cell death. So it sounds counterintuitive to have sort of death of cells being part of a process, but in embryogenesis it is part of the normal process. Yes, I'm just making sure that I haven't left anything out. So this is just a schematic of what it looks like. And then the next image, this is the same stage, but it has been rotated. And this allows you to see more easily the neuroectoderm and how it has formed the head folds. And these are going to form the completed neural tube, but because it's in the head, it will elaborate into a larger structure that's going to form the brain. The amniotic cavity is shown here, and you can, shown here. You can see it, and the amnion, the membrane that encloses it. Eventually, this cavity is going to have to kind of surround the whole embryo, so some rearrangement will be required. And then there's a process here called the allantois. And if you remember the allantois from the chick, that was your gas exchange and your nutritional. No, that was just the gas exchange, because the allantois and the yolk sac were separated. But in the case of the mouse, this is going to form the connection with the placenta. Okay, and you can see it from this angle, but you couldn't from the previous angle. And so I know that it can be hard to sort of figure this out, but just remember that it's a cup. It's a three-dimensional structure, and we're just slicing through it on a slightly different angle. And this is just helpful for you to see kind of a different view so you can understand a little bit about the three-dimensional structure. So this is a couple of days later, well, a day later. And you can see that there's been a lot of development. So those neural folds, all the stuff around the outside has been picked away. So you can't see, for example, the chorion or the amnion or any of those structures. You can see the allantois. It's a bit clearer because the other stuff has been picked away. And this is shows you the elaboration of the head folds. Here's the developing eye. Mesoderm structures are shown in this kind of pale brown to distinguish them from the notochord, which is shown here in red, and the somites that are also shown in red here. And the somites are starting to form. The gut has also started to form. So you can see the hind gut here and the foregut, and they are not, they're somewhat continuous, but they haven't sort of opened at the mouth or the posterior yet. And you can actually, there is a place, here's where the little heart is going to form, and there's a little bulge of mesoderm there that's going to form. So basically this is that embryo before turning. By about stage 9, turning occurs, so that now the embryo is curled around. And now let's just look at this again. So just to point out, like, this is the dorsal part of the embryo, and this is the ventral, and this is the ventral. Anterior here and posterior there. So to rearrange it, this has to be kind of on the outside, and the ventral has to be kind of, back versus belly. And that's what turning accomplishes. So don't worry about the movement. There is a figure in the text, 3.24 or 3.26. And I didn't include it because I don't see the value in memorizing those movements, just so that things flip around. And in addition to the fact that we now have, like, your dorsal back and your ventral kind of normal, the embryo is curled around as they are at this stage. But at this point too, the amniotic cavity is surrounding the embryo, and the umbilical connection is sort of in here somewhere and is connecting to the placenta. So basically, gastrulation is finished, neurulation is complete. You have a neural tube. It's not completely developed yet, but you have a forebrain and a midbrain, and you will eventually get a little hindbrain here. Each of those are devoted to different parts of the brain. And then we have the dorsal and ventral set up properly, and the amniotic cavity around the embryo. Okay, so that's all I want to talk about for mice. For mouse, we're going to talk about the development again when we talk about somites and neural tube patterning and a lot of that stuff later on in subsequent chapters. So this, remember, was just the beginning, the intro. So we're going to just look briefly at the human life cycle. I think you must be most familiar with the human life cycle, but just like other organisms, there is a fertilized egg. Fertilization, of course, is internal. There are cleavage stages that leads to a blastocyst. The blastocyst looks awfully like the mouse blastocyst. It forms a day and a half or so later. Implantation is a few days later, and then from implantation onwards, everything is much later. It takes much longer for humans than for mice. And the implanted embryo, if you look at it, if you look at this part of it, it turns out that the actual blastocyst part, it's called a blastocyst because it looks more like the chick blastocyst, or the chick blastoderm, sorry, than the mouse. It's flat. We don't have that cup-shaped structure, and so we don't have the turning, the kind of inside out of the embryo. But there's gastrulation. There is folding, so instead of being flat, you have to curve the cells around to produce a three-dimensional rounded structure, and the embryo does curl around this way while it's developing. And after about 4 to 8 weeks, when we get to the fetus stage, at the 8-week stage, much of it, there is still development, but a lot of it is sort of growth, and the maturation of the organs. So you can't be born at this stage because your lungs aren't ready to breathe, for example. But although these days they can do more and more interventions for things like that. Um, let's see. I think that's probably all we need. So the key things is that much of the development that we're going to look at here is going to look very, very similar to what we saw in the mouse. Um, but there is a little bit of a difference with the actual shape of the epiblast and just a few details. It's also a much longer process. So if we look at these early stages, this is a schematic. There's a diagram, figure 339, in your textbook, which you can also look at. It shows what the real things look like. I find these schematics are helpful. You have fertilized embryo. You have the first cell division occurs earlier than the mouse, but then the mouse goes much faster after that. The 4-cell stage, the 8 to 16 cell stage forming the morula, you still have compaction. It's a little later, of course, in the human versus the mouse. Cavitation. So there's a division that separates from the morula into outer cells and inner cells. And then there's cavitation where, remember, the water and the salt are pumped into the space between. And because of the tight junctions between these cells, the water can't escape. And so you wind up with this fluid-filled cavity. All of this is pretty much the same as in the mouse embryo. And we have that inner cell mass and um, uh, the hypoblast in chick, which we call the primitive endoderm in the mouse and in the human embryo. So up to this implantation stage, they look much the same. Okay. But the gastrulation actually looks a bit different. This is an image of the little human embryo. Implantation has taken place, so it's surrounded by basically the uterine wall. The extraembryonic mesoderm surrounds the whole embryo and forms the attachment um, to the uterus to make the placenta. The amniotic cavity is formed right above the epiblast, and the epiblast and the hypoblast are very much like in the chick embryo with a single layer of cells. So you're not seeing that cup shape that we saw. And then if we do a cutaway, and so we look at the embryo, this doesn't show us very much. It's hard to see things except just the general arrangement of the cells. But if you cut away all the surrounding tissue and just look inward, you can see the primitive streak forming, and it starts in the posterior and moves towards the anterior, just like in the mouse and the chick. In this case, it is a furrow. So it's not a thickening of cells, it's a furrow, and it looks more like the chick embryo actually than the mouse embryo. And then when it reaches the maximum distance, there is a visible node. You can see it. The cells actually look a bit different, but they aren't always the same cells throughout the development. But just like in the mouse at this point, the node kind of stays in place and the cells move forward. Okay. And ahead of the node, the head process and that sort of thing is formed. The notochord is underneath, etc. So that, aside from the physical appearance of the primitive streak, it looks a lot like how it works in the mouse. And so we have this flat embryo kind of floating above this big yolk sac. And again, the yolk sac it doesn't have any yolk, or if it does, it's very, very little. But it is at the beginning of development. It is continuous with the endoderm. and it will eventually be reduced until it's completely gone once the gut is fully formed. Okay. And so the first thing that starts happening, this is around day 21, you want to change from that flat disc into a three-dimensional, more embryonic looking structure. And so what happens is there is lateral folding. So the cells move around to the side to form more of a rounded structure, which you can probably see a little bit here. And they also extend in both directions, anterior and posterior, and the embryo sort of curls around to form that, when you see embryos, they're always curled around with the head forward and down and the tail forward and down. The amniotic cavity also grows larger and extends around. So this is the difference between these two, is just a day, just 24 hours, and you can see a big change that's happened already. And then in the remaining few days, like approximately a week later, you see the same changes, but they continue more. So that lateral folding is still taking place, but there's a lot of extension. So the neural tube and the brain and the anterior part is all extending and curling around. And the anterior portion of it, you can see it's widening out and getting larger and forming an actual brain. Whereas in the more posterior region, it is an actual neural tube. The gut, you can see the gut here, the foregut and the hindgut, it hasn't broken through to form a mouth yet, or the anus on the posterior side, but the yolk sac is getting proportionately smaller because it's getting shrunk in, and it will eventually disappear once the gut is fully formed. At this day 28, just 4 weeks, there is you can see the heart, and there's a little stomach that of course isn't complete yet, but is starting to form, and so on. And this is the connecting stalk that's going to move around and connect into this region. And this connecting stalk is what connects the embryo to the placenta. So I want to talk briefly about the placenta. So, one thing I forgot to just mention is that in these pictures, starting from here, there is a chorionic cavity, and that is it's called chorion because it's similar to the chorion, which is lining the eggshell, if you remember, of the chick embryo. But in this case, its function is the placenta. So there's all these other cells that move in, but the chorion is part of the formation of the placenta. So this is kind of how it works. The chorion forms villi, and there are cells on villi are finger-like projections, right? So think of your intestine; it has villi, right? The projections into the gut, into the intestine, and on the surface of these are they're called syncytiotrophoblast cells, and they're similar to those giant trophoblast cells of the mouse. They're called syncytio because they are bigger than the cells normally, and they have multiple nuclei because, once again, they go through the process of mitosis without actually doing the final division. So you've got these really big cells with lots of nuclei, and they form these villi, and the villi are connected to the embryo's artery and veins, but what the actual syncytiotrophoblast cells are doing is they're working their way into the uterine lining, and they are remodeling it so that the arterial cells, or sorry, the arteries are kind of opened in a way, and the veins are kind of opened in such a way that it creates these are like little areas that have they're just pools of maternal blood. So the maternal blood is coming in here, and it's bathing the villi, and we're seeing the exchange of gases and nutrients. So nutrients go in, and they are transported to the umbilical vein, which is the red one. So nutrients pass into the fetal blood system. They're just through diffusion across here. But oxygen also passes. And one of the reasons, is that the fetal hemoglobin molecule is a different one from the one of the adult, and it has a higher oxygen affinity. So what happens is that the adult hemoglobin loses its O2 that gets picked up preferentially, or transferred preferentially to the fetal hemoglobin, in a sense. And so that that difference in affinity is necessary, or else there'd be a lot less oxygen being transferred to the embryo. So that umbilical vein is bringing that oxygenated and nutrient-rich blood into the embryo, feeds the embryo, provides oxygen for the cells and so on, and then the umbilical artery is bringing back the non-oxygenated, nutrient-poor blood to kind of refill, right, to top up again the the oxygen in the food so that can be delivered again. That might be all. I think that's all I need to say. Okay, so for the next lecture, which will be on July the 7th, I will start on chapter 4, but I'm not going to put that on the midterm because the midterm is on that Friday, and it's not enough time to really properly study that material. Unless I hear objections and people would really prefer something else, my plan is to do another two-stage review the lecture before, so on July 9th. If you would rather not, and if a lot of students would rather not, then I'm okay doing, not doing that. But I do think it's useful, and I think students have been commenting that they liked it and they found it valuable. Okay, that is it. Thank you. Don't forget to ask questions if you are needing information.